Okay, Assalamualaikum everyone. Okay, can you hear my voice? Yes. Okay, so as scheduled today, we are going to have our elite fellow to give his lecture. Okay, so the lecture is 1 hour. And I think today he's going to give some lecture on a little bit on flavonoids. Okay, so take this opportunity to ask him questions, or you can just ask him whatever you want, or whatever you want to know, right? Throughout the class, I'll be recording this class. Okay, I'll start recording. Okay, so we have 35 students here. Okay, so I'll pass to Professor Dr. Muhammad Zakaria for his lecture. Okay. So sorry.
Hello, can you hear me?
Yes.
Yes,
Yeah,
Doctor.
good afternoon. So we continue. Today we learn more about the application of flavonoids.
Tekan. Pin. Tekan. Pin.
Hmm.
Pin. Okay.
So we're going to learn more about the application of flavonoids, um, the structure and function of flavonoids. Okay, mana this slide?
Okay, share
Share.
the entire screen. Okay, buka.
Maybe it's a bit soft. One thing.
Okay, can you hear?
Okay, can you see this one?
Yes, doctor.
Yes,
Yeah.
doctor.
Yes, doctor.
Okay, let's see now. Okay. Um, so flavonoids are widely distributed in plant kingdom. So anything that you see in the plant kingdom, in outside your house, in the plant, in the garden, that are colorful, there are two potential substance, class of substance. One is flavonoid, the other one is carotenoids. There are some other class of compound which are colored as well, but these are the major one. Okay, but flavonoid, as we have described earlier in the last lecture, are widely distributed in the plant kingdom, but play vital role as pollinator and as protection for the plant and so on. So they can be found, okay, where? Okay, the second sentence, they can be found in the petals, crops, seeds, leaves, stem, roots, and buds, okay? So it is reddish to brownish, yellowish, all depend on the structure of the molecule. Flavonoid of a fern contain C-meta derivative. Fern is different. So they are different class of plants. Glycoflavonoid can be isolated from seaweed. So that's why when you take seaweed, it's a bit like jelly, a little bit jelly, because of the glycoflavonoid. Inferior plants and mushrooms have them only very rarely have the flavonoid. Microorganism and algae do not produce flavonoid. So these are the structure I have described. Very important to see the C6, C3, and C6. Okay, these are the carbon skeleton. Okay, the carbon skeleton of flavonoid. Um, I cannot move my cursor. Just wait a minute. Try to move my cursor.
Okay.
Huh? Boleh. Oh. Okay.
Tak connected, is it?
Mm-hmm. Okay. Okay, never mind. I couldn't show you my cursor. Okay, if you look at the structure of this flavonoid, there are three different basic structure. The neoflavonoid, which is a minor class of the flavonoid. The isoflavonoid, where the aromatic ring moves from position 2 to position 3, and the normal flavonoids. Okay. So in the biosynthesis of flavonoid, this is play such a vital role because the basic ingredient for the biosynthesis of flavonoid contain basic shikimic acid biosynthesis. We produce phenylalanine, and then they convert it to cinnamate, and then another portion of the flavonoid are derived from acetate polymalonate pathway. There is acetate and two molecule, one molecule acetic acid, two molecule malonic acid from the pyrene ring. Flavonoid acetized by phenylpropionate metabolic pathway with the amino acid phenylalanine is used to produce 4-coumarate-CoA. This can be combined with malonyl-CoA to yield the true backbone of flavonoid, a group or compound called chalcone, which contains two prenyl rings. Conjugate ring closure form the three ring structure of the flavone. Okay, so there is maybe if you look at this diagram which I showed to you, then you can see how the whole process. And then from coumarate cinnamyl CoA reductase convert the coumaric acid into compound called lignin. Lignin are substances that form the structure of wood. Okay, so for the strengthening of the wood or the tree, it was lignin that bind to the structure of the cambium. Here is an additional metabolic pathway. Maybe you can have a look yourself from naringenin. They can produce many different types of flavonoid. So that's why if you get one compound, one flavonoid from a tree, there are many other flavonoid which are related to it. Okay, then you can have a look yourself. Enzymatic conversion of naringenin to apigenin. Naringenin, isoflavone. If you look at the isoflavone, how can I show you the? You look at the pyrene ring here. There is no double bond in the position 2 to 3. Compared to apigenin, there's a double bond position 2 to 3 in the pyrene ring. Because of this double bond, apigenin is colored substances and naringenin is non-colored substances. It's colorless, but they are definitely detectable by UV light. But apigenin can be seen as a color. But the different types of functional group in the position 7, 5, and then in the adjacent aromatic ring, position 3, 4, and 5. This substituent, which could be OH or O-methyl or any other structure, will result in the different shades or different colors of the flavonoids. Here, aromandrin dihydrocampherol. These are some common compound. They can be converted to quercetin, methylated quercetin, and quercetin sulfate, and many different types of molecular structure. And each of these compounds have different biological behavior. Okay. The enzymatic conversion of dihydrocampherol to pelargonidine. Pelargonidine is this, whereby subsequently they are converted into cyanidine. Anthocyanidine are substances that is very, very colored. You see some flowers are blue or dark red, dark orange. There's possibility the colors are due to anthocyanin or anthocyanidine. So now look at the IUPAC nomenclature for flavonoid. You have flavone, 2-phenylchromane 4,1. Then the flavanol, 3-hydroxyflavone, 3-hydroxy, 2-phenylchromane 4,1. Flavanone, 2,3-dihydroquinone 2-phenylchromane for 1, flavanol, 3-hydroxyflavanone, and then on top of that you have other flavonoid, this isoflavone, anthocyanidin, proanthocyanidin, catechin, chalcone, dihydrochalcone, and uron. So the state of oxidation depend on the ring B, A and B, okay? A and B or even C. Anthocyanin are the real flavonoids. These are anthocyanin, benzopyranchromone, and pyrone, gamma pyrone. Natural derivative from benzo gamma pyrone structure are considered as flavonoid by certain researchers. These molecules are anthocyanin. They are classified as another group, anthocyanin. So special flavone like coumarone, also coumaroflavone, and bioflavone, comestan derivative, and rotinoids. Okay, what is interesting in this group of compound? We look at rotinone. Rotinone is basically a very important insecticide at the early 1900. Those days, when they travel by ship for months and months in the ocean, they use rotinones as an insecticide to keep all the dry food, the grains, rice, beans, mixed with rotinoids to protect against insects. So the structure below, rotinone, basically rotinone from a plant found in Southeast Asia. There is pokok tuber, or there is species, or there is elliptica, or there is there is elliptica, there is malacensis, and many other dairy species. This plant contain rotinone. If you have this plant, you can find it very easily in Malaysia, where if you put it in water in the river, the fish will get suffocated, because what happened, the rotinone will block the electron transport chain in the fish through the gills and result difficulty in respiration. So the orang asli, or some people local even today, they use pokok tuber to catch fish in the river. The only problem, if you put the tuber tree in the water, many fish will get killed. Okay, how does it get killed? Because the rotinone block the electron transport chain. Okay, electron transport chain. Electron transport chain are involved in respiration. So flavonoid structure variation, there are so many different variations, but I cannot move my cursor. So you can find their position 3', 4', 3, position 5', F5 and 7. These are places where they can be substituted for OH, OCH3, or any other, or glucose or carbohydrate. As position 3 also can be OCHO and so on. And position 3', 4', and 5' is another position where can be substituted for OH and OCH3, O-methyl. The various substituent results in different color for the structure of flavonoid. Anthocyanidin Anthocyanins are always in the glycoside form, in the vacuole in cell fluid. Catechin and cyanidin can be stored in the aglycone. Aglycone means no sugar, okay? Form. Tannic acid dissolved in the essential oil, okay? The possible role of flavonoid in plant. Flavonoid are secondary metabolites of plants, and they are providing the color and flavor material. So it protects against UV radiation. Okay, the flavonoid protect the plant from UV radiation. There's a lot of UV radiation from the sky, from the sun. If the plant are not protected, the plant can die, okay, of the UV radiation. So the function of the colored substance is to absorb this UV radiation. Second, to protect from fungus and protect from insect and snail or other pests. They are signal for end-bound bacteria. They modify enzyme reaction. So these are some other activity of flavonoid. 75% of angiospermae, flowering plant, contain camphorol, quercetin, and 10% of them are myricetin. Okay, so these are the common flavonoid you can find in plant. So you take any plant, the possibility you get camphorol, quercetin, and myricetin are very high, because they are used by the plant as antibody. Just like us, we have antibody, the plant have this as their antibody. Common flavonoid glycoside, example rutin. Rutin is one very interesting flavonoid. Rutin can be found in 50% of the angiospermae. Quercetin is a glycone form of number of other flavonoid glycoside, such as rutin and quercetin, found in citrus fruit, buckwheat, and onion. So why rutin is so important? Rutin is a compound that can strengthen your blood capillary. You know, some people, if they get hit or somebody tap on the body, the tissue will turn blue because the capillary is injured or damaged. So if you have rutin can help to repair the damaged capillary. So one of the plant that you can find is that contain a lot of rutin is pokok gambe. Tahu pokok gambe? Uncaria gambia. Uncaria. Uncaria gambia. Gambe. Gam bia. Gambia. So they are used by Malay, Indians, and people who take sirih as part of their addition. So gambe, they use for eating it with sirih because it can cause slightly bitter and astringent, but it helps. That's why for people who take sirih with gambe, their blood vessels are strong and their capillary are strong as well. Bioflavonoid have bioactivity. For example, high concentration of quercetin in vitro experiment causes DNA mutation. High concentration of quercetin in vitro experiment causes DNA mutation. In spite of that, epidemiological study declared that this flavonoid consuming prevent atherosclerosis, infrared, and infarct, and other disease. Just I mentioned earlier, it can help to strengthen the vessels. So there's also some potential hazard taking too high concentration of quercetin. Flavones, flavone. So these are the where you can find flavone in the sayur, some sawi, and some kutumba, uh, kutumba in wheat. But flavones are very important substances where they have demonstrated this flavone, when taken in regularly, help to strengthen our immune system. So if you take this flavone, there are demonstration that it helps to strengthen their immune system. So the flavone have got immunomodulatory action. Flavone have got immunomodulatory action or immunomodulator. Is a substance called immunomodulator. So that's why our whenever taking vegetable like essential for man and animal. So naturally, we cannot run away from plants, as plants provide the necessity substances to protect ourselves from infection. So for example now, period of the COVID, then you should take a lot of vegetable that's if they are green or they are colors, you take. So that's why you see the monkey on the tree, plenty of monkey behind the Faculty of Science. They eat flower, and this flower contains flavone. That's why the monkey are very strong. There's seldom get infection because the flavone help to strengthen their immune system. We strengthen our immune system by taking vaccine, but monkey take flavones. Okay, these are the natural way of protecting themselves. Flavonoids. So just now we talk about flavone. You see that structure, flavanone, where the double bond at the 2,3 position removed. So definitely this become non-colored substance. Okay, non-colored substance. Recent in vitro examination strengthened the anti-proliferative effect of the prenalated naringenin. This is from Humulus lupulus on prostate tumor cell line culture. So they have anti-cancer property. The flavonoids, they are found in the here. If you look at in these oranges and the grapefruits, where the flavonoids are, they are found in the skin of the inside the skin of these oranges. Okay, not in the not on the fruit content. Okay, in the skin, not on the flesh. But definitely the color yellow in orange and grapefruits, or even here limau, the limau, the color yellow. The yellow color is from carotenoids, another class compound. Okay, but there are also flavonoid. So if you want to improve, strengthen your antibody against cancer, then you should take this basically to take the skin, the skin of these fruits. Okay, flavanol, medicinal plants. Okay, taxifolin. This is another anti-cancer drugs substance that is found. But we always take the rind, okay, the flesh, so we lose the value of the fruits. Okay, so anthocyanin, you see the in the grape in orange, the grape, the blue, dark blue. Also, if you look at roselle, okay, these colors are due to another class of compound called anthocyanidin in the brinjal, okay, inside in the cherry. So in the wheat, okay. So sour cherry, blue grape, plumberry, red cabbage, egg fruits, cereal, elder, sop tree, and many medicinal plants contain anthocyanidins, maybe in the class of plagonidin, cyanidin, or diphenidine, depend of the functionality. It is important, it is very good if you have, they have found this anthocyanidin when taken in regularly help to reduce the blood pressure. So people with hypertensive hypertension, they can take some discolored fruits to reduce the blood pressure. Isoflavone, these are medicinal plant, leguminous plant, or soya bean. It contain genistein, daisine, or biocanin. So beside that, inside this plant, soya bean, it contain also catechin, and they are important. These are isoflavonoids. They are important to give antibacterial, antifungal activity. So phenolic antioxidant can function as a free radical scavenger and a metal chelator. Flavonoids are highly effective scavenger of all type of oxidizing radical. Rate constant of quercetin with superoxide anion is 0.9 times 10 to the power 5 at the pH of 7.5. The arise phenoxy radical relatively stable molecule. So the molecules help to cause degradation of hydroperoxide. Okay, these free radical have to break down hydroperoxide. Hydroperoxide are substances produced by the body metabolism that can cause injury to the tissue. So body tissue can get injured when the body produces a lot of hydroperoxide. So taking some of these flavonoid, it has the free radical scavenger activity that that destroy this peroxide. Okay. Chelating ability of flavone and flavanone. Okay, so if you look at the this nearby two oxygen functional group bind to copper, and these are important of getting rid of metal ion from the body. Yeah. So if there's too much metallic substance in the body, not good as well. So the function of the flavonoids, such as hydroxyflavone, hydroxy, uh, hydroxyflavone, acid flavonone, whereby they cause chelation. You see the oxygen bind to the two oxygen group, and as the as the substance get removed, the copper is eliminated. But in some other example, presence of some of these simple cationic substances has the ability to act as bacterial inhibitor. So ionic copper has also ability to inhibit activity of bacteria that is found in the plasma. These are mesomer structure of quercetin hydroxide radicals, condition of antioxidant property. Phenolic group can easily give a hydrogen atom. Okay, maybe you can have a look yourself. The physiological activity of flavonoid. Flavonoid have been referred to as natural biological response modifier because of strong experimental evidence of their inherent ability to modify the body reaction. Antioxidant or scavenger activity. So the other effect is immunomodulating, which I have already explained to you, as well as anti-inflammatory effect, which I later described to you. Anti-asthmatic, anti-allergic effect, antiviral, antibacterial, antifungal effect. And some flavonoid, especially the flavone or isoflavonoid here, they have estrogen activity. So you know that estrogen or hormones very important for the body metabolism and also for skin care and body activity. So if you have some rashes on your skin, you can prescribe lotion contain estrogen. But the only problem, if you use the normal estrogen, which are based on a different structure that is derived from terpenoid, it has side effect. Prolonged use it causes hardening of the skin. But if you use estrogen, there is activity from isoflavonoid, it's no side effect. So that's why this type of isoflavonoid are very good at making cream to protect skin from rashes. Okay, and also if you take it, improve your estrogen activity. The effect of mutagenicity and carcinogenicity. So flavonoid can reduce potential cancer. Hepatoprotective effect, and it helps to protect the liver from damage. Effect on circulation, modification of permeability of blood circulation. It's improved the vascular structure of the vessels. So can you see the various activity? Uh, one example of flavonoid silibinin. Yeah? Silibinin increases synthesis of RNA polymerase. Okay, helps improve cell production. So these are how cell uh, are produced. So you have more RNA polymerase, that means faster the cell are being produced, duplicate. So if you have a fast growing cell, then it helps to improve the, your skin structure. That's why sometimes you have some cream which contain silibinin. You apply, then your skin after a while look very beautiful, very firm, because the ability to produce more cell quickly, because our cell continuously being damaged and continuously being produced. But as we get older, the amount of removed cell is retained, so you go into aging. Okay, so if you use some of these flavonoid, for example silibinin, on your tissue, then your tissue get improve, get become more firm. Okay, catechol type antioxidant was used in case of alcoholic liver disease. People will take catechol. At one time, they use this cyanidinol 3 to take care of the liver as antioxidant for damaged liver. But somehow they have been banned because of some other problem associated. It was also available in Hungary but became banned because it caused hemolytic anemia. Metabolism flavonoid depend on several factors. 1% original form of flavonoid can get into circulation, structure oxidation, rate of glycosylation, conjugation property, rate of polymerization. These are flavonoid depend on several factors. Okay, metabolism. That means flavonoid after we take, they need to be removed to the kidney. But to remove the flavonoid, they had to be converted into various structure by many ways, by glycosylation, conjugated to other things, rate of polymerization, antioxidant property, absorption, and have bacterial enzyme activity effect on the derivative are often different from natural molecule. Only 1% of the original flavonoid can be get into circulation. Okay, not much of the flavonoid can be absorbed, but when they absorb, they play a vital role in the body metabolism. What happened with flavonoid in our system? Flavonoid are extensively metabolized, and the metabolite arise in intestinal absorption. Flavonoid are transformed into phenolic acid by enzyme colonize gut microbes. These phenolic acid are also absorbed and metabolized in the liver. Generally, flavonoid may undergo conjugation, oxidation, or transformation by cytochrome P450. Glucuronide and sulfate conjugate are formed with majority of flavonoid in the small intestine and in the liver. Why small intestine in liver? Because the antioxidant property. So that's why in our intestine, flavonoids play very important role. Intestine are very active tissue division. So if not enough oxidation, some of the tissue can become mutated or they can become cancerous. So taking flavonoid help to remove as much of the potential factor that can cause cancer. Here, biotransformation of flavonoid in the microsome, in the cell. The liver is the main detoxificating tissue. So liver is very important. If liver fail or liver function fail, that means you are going to say bye-bye. Okay? Where the elimination of isogenic toxic agent happen, mixed function oxidase are localized in the endoplasmic reticulum, and they can catalyze a lot of oxidation reaction, and they have lipid peroxidation end products. So these are example, naringenin from iridocycloid by liver microsome. Okay, there's another one, chrysin to epigenin. So they make the substance become more water soluble through the oxidation. Luteolin. In the old days, luteolin, the yellow substance found in plant, used as coloring agent, especially in Europe. But I think they are probably still used in some making a wine and so on. Transformation luteolin by bacteria and microsome, flavonoid glycoside can undergo changes because the effect of bacteria enzyme colonize in the bowels, in the terminal ileum and colons. Mixed function monooxygenases are able to modify flavonoid and improve, I think. Improve the skin structure. Luteolin 7O glycoside convert to luteolin. Luteolin then inside the liver they convert to luteolin glucuronide, going back to luteolin. Plasma level of flavonoid, if we look here, quercetin, you take quercetin, catechin, methoxycatechin, diosmin, and rutin. In milligram, 65, 32, 30, 10, and 1.4. Where the microgram is less than 0.1. So small quantity, but very important for the maintenance of the oxidation state of the plasma. Regulation of cells depends on redox hemostasis. Flavonoid can influence the redox hemostasis in several steps. In prostaglandin biosynthesis, in leukotriene biosynthesis, in FFKB translocation to the nucleus, in PKC activity, in PI3 kinase activity, in signal transduction, and in cyclic AMP activity in extracellular space in tumor. We can simply say that free radicals in small concentration may cause apoptosis, in large quantity necrosis, tissue death, or that's non-reversible. So the body needs a small quantity of these substances. So here is the biosynthesis of prostaglandin. Camphorol inhibits cyclooxygenase enzyme. So when you inhibit the cyclooxygenase 2 enzyme, that reduce inflammation. Some people got inflammation for no reason. Suddenly they get body become heated up or swollen because of the activity of prostaglandin or enzyme cyclooxygenase. Okay, so some of these flavonoid ability to inhibit or cause anti-inflammatory activity. Leukotriene, quercetin inhibit activity of lipooxygenase. Therefore, they have anti-inflammatory properties. Prenalated flavonoid can inhibit 15 lipooxygenase activity. So leukotriene activity, prostaglandin activity, metabolism that are found in the cell in our body, they cause inflammation, but they have other function. Okay, but to reduce the inflammatory activity, so the flavonoid play such a vital role, like quercetin. Okay, flavonoid and signal transduction. So the polyphenol-like compound inhibit signal transduction pathway, TNF alpha mediated activation of NF-kB pathway, beta kinase, and IL-1 beta activated NF-kB pathway, which is partly distinct routes. NF-kB activation by IL-1 require IL-1 receptor associated with protein kinase activity. So when they inhibit these, when flavonoid inhibit some of these function in the cyclooxygenase activity, then the result is reduced inflammation of the tissue. So look like such important role in the body. Flavonoid in the active center of P13 kinase. You see here, potential inhibitor of protein kinases, glycosylation and glycol moderate the inhibitory effect, small molecule alteration, and strengthen the inhibitory activity and selective activity, hard to predict. So flavonoid in signal transduction, effect of flavonoid. So proliferation and spreading of tumor depend on the activity of the matrix metalloproteinases. Matrix metalloproteinases help to reduce tumor activity. In the formation of extracellular in the matrix, collagens take part in the delimitation individual cell population. Zinc contain matrix metalloproteinases do continuous renewal of the extracellular matrix. Tissue inhibitor and alpha-2 microbulin inhibit MM2 ACEs. Okay, so flavonoid could also induce that help kill cancer activity, invasion. So the function of the matrix metalloproteinases is degradation of extracellular matrix, tissue remodeling, growth, angiogenesis, and metastasis. So flavonoid play a vital role to hold the structure of metal in the protein, protein in the body. Here, some example, matrix of metalloproteinase, flavonoid and signal transduction, data according flavonoid. Okay. Flavonoid are substances from plant kingdom essential for healthy life of man and animal. Thank you for your attention.
Okay, thank you very much, Prof, for your lecture today. Does anyone have questions to ask? Anybody have questions? Do you have a
Any
question?
question?
You can ask any questions, any questions you want.
Otherwise, you go through the lecture, try to understand them, because if I can see you face to face, I can explain better. Those days, it was in the classroom.
Mm.
Today, we are only avoiding the situation of disease. So if you take a lot of flavonoid, maybe you don't get uh, COVID, and you can have class together in the classroom.
Yeah. So there is so meaning compounds from this natural products from plants are actually very useful for us. Okay, so I hope that everyone understand today's lecture on flavonoids and try to understand more on the the functions of this secondary metabolite. So if you don't have any questions, we shall stop our class here today. Don't forget to take your attendance, and so we'll hope to see our elite fellow for another, I think, two more classes. All right, so if you have no questions, thank you, Prof, for giving us lecture today. So we'll see you next time, and thank you all of you for attending today's class. That's all from me. Thank you.
So happy New Year to everybody. Hopefully this year you drink a lot of juice, and take a lot of vegetable. Don't forget that. How important are plants to us? Bye bye.
All right, see you.
Thank
Thank
you,
you,
Prof.
Doctor.
Thank you,
Thank
Thank
you,
you.
doctor.
Prof.
Thank you,
Thank
Thank
you,
you,
Prof.
doctor.
doctor.
Happy
Thank
New
you,
Year.
Prof. Thank
Thank
you,
you,
Doctor.
Prof.
Happy New
Happy New
Year.
Year.
See you guys.
Happy New
Happy
Year.
New Year.